My name is Fabio Fiorani, and I'm presenting highlights of results obtained in a research project focusing on phenotypic diversity in Chilean quinoa germplasm. Quinoa is a species originated from the Andean regions in South America and is adapted to harsh environmental conditions, especially low water availability, temperature, and salt. And it produces very good seeds, and they are nutritionally balanced. And the reference genome has been sequenced, and more sequencing is ongoing. However, to date, phenotypic and genetic diversity remains largely unexplored. In this research project, we focused on the Chilean coastal lowland biome and identified a collection of about 100 ecotypes representing variation within this biome. One of the main reasons of focusing on this biome is that most of the accessions are day neutral concerning flowering time, and therefore are also potentially adaptable to a more northern latitude. In greenhouse experiments, we could characterize these 100 plus accessions, and in the principal component analysis displayed in this panel here, you can see that morphological yield and flowering time traits, days to flower and days to maturity, they cluster in 4 distinct groups, as shown by the clustering here below and by the coloring in the PCA panel. And overall, we can conclude that there exists in the yield and morphological parameters, there exists a sufficient variation for further analysis. One issue that exists in seed processing in quinoa is that the seed pericarp contains a relatively high level of saponins, which are 3-terpenoid glycosides, which need to be removed before human consumption because they confer a bitter taste. We therefore analyzed in the lab the saponin content of our 100 accessions panel and found out that most of the lines actually have a relatively low saponin content, whereas only a smaller fraction of lines can be classified with high saponins. This is actually good news in view of the future potential utilization of some of these lines for pre-breeding efforts. We conducted a field experiments in the southern Atacama Desert comparing 9 selected ecotypes from the 100 accessions panel and one commercial variety which is called Regalona. And our experiment looked like this. We had a reduced irrigation split plot design and a full irrigation split plot design. In these panels on the right, you can see the environmental conditions during the experiment. I want to highlight that under the conditions of the experimental field station of INEA in Chile, in the southern Atacama Desert, there was no rain during the experiment, and the treatments of full irrigation and reduced irrigation were actually as intended, taking place from the initiation of flowering until seed filling. A principal component analysis reveals that in this field experiment, the 2 treatments could be well differentiated. Here, the purple cluster and the green cluster, and in both cases the reference variety, commercial variety Regalona, actually is well separated from the other genotypes, and some genotypes like this CLS9 is actually closer to Regalona. If we then look at some individual traits explaining variation along the principal components axis, I would like to highlight only the ones that we acquired using portable instruments or imaging instruments, and in particular I would like to draw your attention on a thermal index that we calculated, which is, and I jump to the right part of the graph, then has moderate correlations with some yield parameters. In contrast, the photosynthesis as measured as PS2 efficiency at low and high light is actually doesn't have a big explanatory power or contribution to the PCA, and also if you look at the correlations, they are actually absent. We also measured spectral reflectance with a handheld spectral camera at the plot level. In this figure to the left, you see spectral indices to the right, a box containing all correlations with pairwise comparisons between genotypes and treatments, and within treatments and genotypes for each comparison. What we found is that the red-green index and the modified red-green index had most explanatory power for several of these comparisons, indicating that they have actually the potential to be used for further characterization of genotypes of quinoa in the field. In conclusion, we saw that there are good morphophysiological and seed composition variation in the Chilean coastal lowland germplasm, that some imaging techniques show a good potential for high throughput field phenotyping in future studies, and that the diversity in Chilean quinoa germplasm can be exploited in pre-breeding and breeding programs. To this extent, I would like to show you a preliminary plot of genome-wide association mapping showing that indeed there are some chromosomal peaks that we need to explore further. I would like to acknowledge the German teams working in this project in Forschungszentrum Jülich and RWTH Aachen, and also our collaborators in INEA in Chile, and the Ministry of Research for funding.